All right, let's talk about genes that increase due to coronavirus, CXCL10. I don't want to run out of time, and I want to leave some time for questions, so I'm going to kind of go through this fairly quickly. I believe this was the top 25 genes that with the greatest average change. Again, here are genes here. Here are the different time points, day post-infection. Here are our normalized fold change values. So again, it goes from 100 to a negative 100. So in this one, it had the highest values over all the different time points if we averaged all these out, right? When you take all these genes, you can basically put them through string. I saw a lot of string diagrams today, which is awesome. I love string. But again, we can see specific biological groups come up, and they're given here on the right. But what I want to do is point out, so the CXCL10 seems to be connecting to a lot of this stuff. CXCL10 is a chemokine. It's also known as interferon gamma-induced protein 10, IP10. What it does is when a cell gets infected, it basically, shoots out this cytokine into, to the extracellular environment as an SOS saying, help me. And it eventually finds a CXCR3 receptor on an immune cell and basically signals that to basically, to secrete interferons, but also, to basically come help itself. So in a way, this is a gene that is screaming out, I need help, I'm infected. Let's go get the troops to come in and take care of this. What I really like to do when you find an interesting gene, figure out where it's mostly expressed. And that's what we did with the CXCL10. What we found was, the highest expression was in lung, which makes sense for COVID-19, right? This virus affects lung tissue. You have a high level of it in there, which maybe makes sense. Maybe this is in preparation. The lungs are particularly vulnerable to viral infection. What I also thought was very interesting with this correlation, base-based correlation program with Illumina, what you can do is put in a gene and you could say, give me all the diseases where this gene is upregulated. Or actually, not only can you give me all the genes where diseases where it's upregulated, but rank those. And as you can see, these are the diseases it's overexpressed in, some of them, the top ones, and you'll notice all of them are viral. So absolutely this gene, we feel is very important in viral infection. And not only us, but, again, it's nice you do this research and then people also do their own research, and absolutely this chemokine or cytokine is absolutely important in lots, in severe SARS-CoV or COVID-19. Lots of information on that. So again, relating the mouse data to humans, what we find that's really hot and, induced in the mouse, we also find in the human. So what I find, though, is it's sometimes more interesting to look at the genes that actually decrease in expression. So this is the BEX2 gene. So again, same kind of graph here. BEX2 had the greatest average decrease in expression with infection. We can kind of graph that out. Not a lot of connections, not as much as the genes that get induced. But again, BEX2 is our top one. We're also seeing this BEX4, but I also mentioned there's another family member called BEX, that's BEX1. That was also just missed this cutoff just a little bit, but it's also reduced as well. Again, here's the BEX2. What was interesting is we start looking up, you can look at the expression of this BEX2. Again, this is a graph here. This is day post-infection. These are different viral doses. And as you can see, it's almost like a clock, this BEX2, how it works, right? The more initial virus you have, the quicker it goes down. And as you back off that, it doesn't go down as quick. So it's almost like a really good time signature for when that initial infection happens, right? Also, what we found is there's absolutely an age effect. So again, it seems to be, and it's not just this gene, and I don't have time to show that data, but it seems like the older individuals, which are in red, seem to sense this virus or react to the virus much quicker than the young. And again, remember, the young survive this disease or respond much better than the older individuals do. Again, we had no idea what BEX2 does. Started looking it up. I found a bunch of cancer, maybe some cancer stuff, uh, apoptosis, but really nothing viral related. Again, you can look up what diseases are BEX2, the highest, where do we see the greatest change in what diseases? And again, lots and lots of infection studies, especially viral, especially lung. So again, even though I don't see a lot of data or papers out there on BEX2, based on how it reacts with these other diseases and the software I'm using, yeah, it's absolutely probably associated with viral infection. What's probably happening is the virus is probably reducing or inhibiting this gene, right? It's trying to get rid of the COP. So it's probably serving a protective effect. What I find interesting, again, it's X-linked. So it's on the X chromosome. What that means is females have 2 copies of this gene. And I looked at it, it's actually in the area where you don't have that condensation with the bar bodies. So females theoretically should be able to produce both copies of this gene, this protective enzyme. And why does that matter? Is because there is absolutely a gender effect with COVID. Women are much better able to respond to the virus than men do. So could it be that this BEX2 gene, because women have 2 copies, makes them more resistant to the virus? I also found, as you look through stuff, you find all kinds of interesting things. But BEX2 actually binds to olfactory marker proteins. So maybe it's associated with the olfactory system. Virus comes in, knocks BEX2 down. Maybe you're altering the olfactory system, and therefore maybe that's why you lose the sense of smell. I don't know. Can't prove it yet, but I think it's very fascinating and kind of an interesting question.